Bismillah Rahman Rahim, everyone. Welcome to Ordinary Days, and today we are going to discuss the second portion, that is the energy generating phase of our glycolysis. So, now it has all these steps. Till now, if you have watched my previous video, I told you that what will happen in the last step is that dihydroxyacetone phosphate via the triose phosphate isomerase enzyme, it will be converted into this glyceraldehyde 3 phosphate. So as a result, we will get 2 glyceraldehyde 3 phosphate. That is why I have written 2 with it. So every reaction that is happening from the glyceraldehyde 3 phosphate to pyruvate, it will take place twice, because we have now 2 molecules of this enzyme, this substrate. Now this substrate, the glyceraldehyde 3 phosphate, will be converted into 1,3 bis phosphoglycerate. So what actually happened over here? The main thing that happened over here is that apart from this third position, a phosphate group is attached to now the first position. So now the first and third position of this phosphoglycerate are having the phosphate group attached. That is why 1,3 bis phosphoglycerate is the name. Now, because another phosphate is added over here, this phosphate is an inorganic phosphate that has been added, and this reaction is catalyzed by the glyceraldehyde 3 phosphate dehydrogenase. The enzyme is dehydrogenase, so this enzyme is going to remove the hydrogen. So with what thing is there that will take the hydrogen? We have readily available NAD plus. So this NAD plus will come, it will take the hydrogen, and where will we get the inorganic phosphate from? It will be a separately placed inorganic phosphate that will be that will be present in the surrounding here and there, and we will get that, and we will add it to it. So we will get this 1,3 bis phosphoglycerate, and we got the hydrogen removed by the dehydrogenase enzyme, taken up by NAD plus. So simple is that. If you look at these notes that I made from the book Lippincott, so you can see over here that this reaction, it is the first oxidation reduction reaction of glycolysis. And as you can see, we got NAD plus, but this NAD plus is limited in amount, and to get this more of this NAD plus, we require reoxidation. This reoxidation of NADH that was now produced, how we will get it reoxidized? We can get it reoxidized by two means: either by the pyruvate to lactate convergence. Whenever we do this, we get the NAD plus regenerated. Or second thing is by electron transport chain. Now they're saying that electron transport chain occurs in mitochondria, and the NADH cannot enter the mitochondria. So how does it go into the mitochondria? It can go via the malate aspartate shuttle or the glycerol 3 phosphate shuttle. So major thing that they wanted to say over here was this NAD plus that is now produced, we can reconvert it back into NAD plus by this reaction, or either by pumping it to electron transport chain. Now the major thing over here is that the first step by glyceraldehyde 3 phosphate dehydrogenase has now been completed. The hydrogen was removed, the phosphate was added. Second thing that they want to say over here is that a high energy bond is created, and when this bond will further go on in the reaction, it will be able to produce ATP. Now the second thing that occurs over here is that this 1,3 bis phosphoglycerate, it will be converted into this 3 phosphoglycerate. The phosphate that is added to this first position, now it will be removed. And which enzyme will do this? Major thing, the phosphoglycerate kinase. This phosphoglycerate kinase is very important, because why does it have the name kinase? It has the name kinase because ATP is the produced. So this reaction from 1,3 bis phosphoglycerate to 3 phosphoglycerate is the first reaction generating ATP in this glycolysis. That is why it is energy generating phase. So ATP has been produced now. And how does ATP get produced in this? Not by electron transport chain, but by substrate level phosphorylation. So this was the whole mechanism. So if you look over here, we get this 3 phosphoglycerate synthesis. ATP is formed by substrate level phosphorylation. That energy for ATP formation comes from substrate instead of the electron transport chain. Now, very important thing over here is arsenic poisoning. It can be very, very important, and when you are in clinical practice, you can have arsenic poisoning patient. So let's discuss all of that. So arsenite, which is which can inhibit the pyruvate dehydrogenase complex. We will discuss that later, but till now remember that the pyruvate dehydrogenase complex can be inhibited by arsenite, and its pentavalent compound can prevent the production of ATP and NADH by without inhibiting the pathway itself. Without inhibiting the glycolysis pathway, this arsenate can prevent the production of ATP that we just saw right now, this ATP and this NADH. It can prevent the production of this. So this arsenic now it competes with inorganic phosphate as a substrate for glyceraldehyde 3-phosphate. So this arsenic it will now do competitive type of relationship with this inorganic phosphate. So if arsenic is more in amount, so it will attach to this glyceraldehyde 3-phosphate dehydrogenase. And what will happen in such a situation? When the arsenate is there instead of the inorganic phosphate, what will happen? The glyceraldehyde 3-phosphate will now be converted into a complex that will hydrolyze into 3-phosphoglycerate. But in this mechanism, the 1,3-bis phosphoglycerate will not be formed. When arsenate is there, 1,3-bis phosphoglycerate will not be formed. When it is not formed, no NADH is generated, no ATP is generated. So the pathway is not itself inhibited, the pathway goes on, but one step is skipped, and this step is major one because it can produce NADH and ATP. So arsenate is so bad in this sense. No 1,3-bis phosphoglycerate is formed, no NADH formed, as you can say, no ATP formed. So this is how it takes place. But now, how does it compete with inorganic phosphate? That firstly, it goes to this ATP synthase. ATP synthase is F1 domain. At the F1 domain, we get inorganic phosphate. So this arsenate competes with inorganic phosphate to attach to this F1 domain. If inorganic phosphate was gonna come and attach to this F1 phosphate, we will get normal ATP. But when this arsenate comes, we get ADP arsenate, which is abnormal. And when things are abnormal, no ATP produced, no NADH produced, no 1,3-bis phosphoglycerate produced, this glyceraldehyde 3-phosphate directly converts into 3-phosphoglycerate. So this is how arsenic poisoning skips the step that we just studied. So this was one major thing about arsenic poisoning. So till now we are done with glyceraldehyde 3-phosphate being converted into 1,3-bis phosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. Then this 1,3-bis phosphoglycerate gets converted into 3-phosphoglycerate by the phosphoglycerate kinase enzyme. Now we are going to convert this 3-phosphoglycerate into 2-phosphoglycerate by the phosphoglycerate mutase. This is just simple isomerization. Next step is this 2-phosphoglycerate gets converted into phosphoenol pyruvate. Now enol thing is happened. How does enol group get formed? By the enzyme enolase. So enolase is over there and it is a typical dehydration reaction, and enolase, the enol forming compound or enol forming enzyme, it is itself inhibited by fluoride. One important thing about this dehydration that was taken by or was catalyzed by enolase is that this dehydration, it is reversible. Also, it is inhibited by fluoride, and whenever you will do the water fluoridation that takes place in certain countries like USA, the lactate production in mouth bacteria is decreased, and it also prevents dental caries. So this lactate production can be decreased by water fluoridation because fluoride, it is going to inhibit the whole pathway, and from here the pyruvate cannot be converted into lactate, and hence this mini kind of water fluoridation, it is going to prevent dental caries. Now the last step are very much important but very simple. The phosphoenol pyruvate gets converted into pyruvate by pyruvate kinase. The enzyme over here, you can also see there is another kinase. So whenever there is kinase in the glycolysis pathway, what do we get? We get ATP produced. So pyruvate kinase produces ATP in this reaction, and this step is catalyzed by fructose 1,6 bisphosphate. What is this fructose 1,6 bisphosphate? Over here, this fructose 1,6 bisphosphate is catalyzing this reaction, or it is supporting this phosphoenol pyruvate to pyruvate reaction. It is known as feed forward type of regulation. So this feed forward regulation, a product that was previously formed in the cycle, it is causing the cycle to keep on going. So this is what feed forward means. Now we can say that covalent regulation in liver takes place also as well. So this is the kind of covalent regulation that takes place in liver. It is very easy. Now let's understand this whole mechanism. What happens is, you know, a major thing that you should know about glycolysis is when carbohydrate is rich, when you have taken a carbohydrate rich meal, you have a large amount of insulin, and that insulin supports the breakdown of glucose or glycolysis. But when you are starving, you don't want your glucose to break down, and hence glucagon comes in action, and hence it inhibits the glycolysis. So when glucagon is in action, it means you are already low on glucose. So glucagon comes to its receptor in the liver cells. The receptor, it catalyzes the adenylyl cyclase. The ATP is converted into cyclic AMP. As you know, cyclic AMP goes to protein kinase A. The protein kinase, what can it do? It can do the phosphorylation. So protein kinase comes over here, goes to pyruvate kinase. The active form is non-phosphorylated, so it converts into phosphorylated or inactive form. It attaches a phosphate to it and shuts down the whole process, and hence the phosphoenol pyruvate cannot get converted into pyruvate. So this is what happened in liver cells when glucagon is more, or you can say when glucose is lesser amount. So we want to shut down the glycolysis process. So this is glycolysis regulation in the liver, or covalent regulation. Now, pyruvate kinase deficiency, it is very important because red blood cells, the mature red blood cells, they depend upon they do not have any kind of mitochondria. So glycolysis is their major source of energy, and it help the ATP that are produced by glycolysis, they help the red blood cell to maintain their biconcave shape and pass through the whole capillaries. But when glycolytic enzymes are deficient, majorly the pyruvate kinase enzyme is deficient, what we get anemia. There is less amount of ATP produced. That less amount of ATP means that the now red blood cells cannot maintain their biconcave shape, changes in the cell shape, and that changed red blood cell, it gets digested or phagocytosed by this mononuclear phagocyte system of our body. Now they are saying that the liver pyruvate kinase and the RBC isozyme, both of them, they are formed from the same genes. But how are they different then? They are different in such a way that they have different start sites in transcription. That is written in your Lippincott. Now, this liver pyruvate kinase, when it is deficient, when this glycolytic enzyme is deficient, it does not have any special effect on the liver, because the liver can generate its own ATP via oxidative phosphorylation or using mitochondria. But when it goes to the red blood cells, as you know, they lack mitochondria, they cannot do oxidative phosphorylation. What will happen? There will be altered kinetics, there will be less stability of the red blood cells, and the red blood cells will undergo hemolytic anemia. Also, a person or a patient who is heterozygous for the pyruvate kinase deficiency, they are resistant to most forms of malaria. So deficiency or anemia in one case is protecting them from different kinds of malaria. So until now, about the energy generating phase, we did the glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate by the dehydrogenase enzyme yielding NADH using inorganic phosphate. 1,3 bisphosphoglycerate gets converted into 3 phosphoglycerate by the phosphoglycerate kinase. This step is very important because it generates ATP. Similarly, the 3,1 gets converted into 2 by a mutase enzyme, phosphoglycerate mutase. This 2 phosphoglycerate gets converted into its enol form by enolase. This phosphoenol pyruvate gets converted into pyruvate, the final product, by the pyruvate kinase. We talked about its deficiency, we talked about its feed forward regulation by this fructose 1,6 bisphosphate. Now we can also talk that ATP itself inhibits this pyruvate kinase. And we also talked about its covalent regulation in the liver one and how its deficiency affects the red blood cells. But one thing that we skipped over here was this 1,3-bisphosphoglycerate. In certain cells, like in red blood cells, it gets converted into 2,3-bisphosphoglycerate. How does it take place? By mutase enzyme. Why? Because we just have to shift the um, phosphate group from one position to the two position. So mutase enzyme is over there. And now when we want to cleave this phosphate at the second position, we will use a phosphatase enzyme. It will use water, remove the phosphate group, and we will get the 3-phosphoglycerate. Now, why is this 2,3-bisphosphoglycerate important, especially for the red blood cells? If you know about your red blood cells, if you have studied your blood module, you know that red blood cells have trace amount. Most cells have trace amount of this compound, but the red blood cells, it is very important because it will help them to increase the oxygen delivery to the cells. With that, we are done with the energy generating phase of glycolysis. One thing that you should know, that every step is occurring twice in this reaction. Every step is occurring twice, and only irreversible step over here is this phosphoenol pyruvate to pyruvate conversion. Only irreversible step in this one is phosphoenol pyruvate to pyruvate conversion. And there are two other ones which are in the first phase, the energy requiring phase. One is the glucose to glucose 6 phosphate one, and other one was this fructose 6 phosphate to fructose 1,6 bisphosphate. So these three reactions, the first reaction, this third reaction, and this last reaction, they are irreversible. All others are reversible. Now the ATPs that are produced, one over here and one over here, they are not two actually, but rather they are four, because we have two amounts of glyceraldehyde 3-phosphate. So we get two from one, so two from other. So if we have glyceraldehyde 3-phosphate and it yields 2 ATP and 1 NADH, and we have 2 glyceraldehyde 3-phosphate, multiply this whole thing by 2, you get 4 ATP and 2 NADH. But these, out of these 4 ATPs, 2 are used in the energy requiring phase. One over here, one over here. So net ATP production is 2 ATP and 2 NADH. Now for these NADH, when they go to the electron transport chain, each of the NADH yields 3 ATPs. So we get a total of 3 2s are 6, that is 6 ATPs from NADH. So this was whole energy generating phase of glycolysis. I hope you understood everything. I did my level best to teach you from the book, from my notes as well. If you understood the arsenic poisoning as well, the pyruvate kinase deficiency, feed forward mechanism, everything that I'm saying, then please like, share, and subscribe to my channel. And stay tuned for more videos coming your way very soon, inshallah.